Good morning, everyone. I am Hana Nafratirova from Department of Community Nutrition, IPB University, Indonesia. Today, I'll be presenting our study titled Mapping the Nutrigenomic Landscape of Okra, Abelmoschusesculentus, a networked biology study of quercetin's anti-diabetic targets. Okra, a widely consumed vegetable, contains quercetin, a bioactive flavonoid with well-documented benefits for metabolic health. Previous studies have shown that quercetin improves glycemic control and reduces inflammation, both of which are critical in diabetes management. Our research builds on this foundation by exploring the nutrigenomic targets of quercetin and its potential mechanism against diabetes. The central aim of our study was to identify gene-level targets of quercetin and to investigate how these targets interact with pathways relevant to diabetes. To achieve this, we employed a bioinformatics-driven approach using protein target prediction and pathway enrichment analysis. To systematically explore quercetin's antidiabetic potential, we combine computational target prediction with pathway analysis and molecular docking. First, protein target prediction was performed using Swiss target prediction and similarity ensemble approach or SEA, yielding 134 candidate gene targets. Next, pathway enrichment analysis was conducted with G-profiler to identify biological relevance and disease associations. Finally, molecular docking simulations were carried out using AutoDocFina. Focusing on the insulin receptor, INSR, and other key proteins. This allowed us to assess binding affinities and compare quercetin's interaction with those of metformin, a standard antidiabetic drugs. Our pathway enrichment analysis revealed three highly significant KEGG pathways directly linked to diabetes. Diabetic cardiomyopathy. This pathway highlights how quercetin may protect against heart complications that often accompany diabetes. Second is insulin resistance. A hallmark of type 2 diabetes, enrichment here indicates quercetin's possible ability to counteract impaired insulin action. Third, insulin signaling pathway. Central to glucose uptake and metabolism. These pathways suggest quercetin's potential role in enhancing insulin sensitivity. Together, this pathway frame quercetin not just as a metabolic regulator, but as a compound with systemic impact on diabetes-related complications. Within this pathway, several key genes emerge as critical nodes in quercetin's network of interactions. By targeting this diverse set of genes, quercetin appears to influence both metabolic and inflammatory pathways, suggesting a dual mechanism of action against diabetes. Our molecular docking simulations using AutoDocFina provided a striking insight. Quercetin and metformin were both predicted to bind at the same site on the insulin receptor, or INSR. This overlap suggests potential competition or modulation of receptor activity, meaning quercetin could either enhance or alter metformin's effects. The binding affinities indicated that quercetin has a strong interaction with INSR, supporting its role as natural modulator of insulin signaling. This finding bridges nutrigenomics with pharmacology, showing that a dietary flavonoids like quercetin may act on the same molecular targets as established drugs, opening the door to complementary or synergistic therapies. In summary, our findings suggest that quercetin exerts anti-diabetic effects by targeting insulin-related signaling pathways and inflammatory responses. This study not only highlights quercetin's therapeutic potential, but also lays the groundwork for further modeling and experimental validation of its gene-level interactions. By mapping the nutrigenomic landscape of okra, We provide new insights into how natural compounds like quercetin may complement existing therapies for diabetes. Thank you for your attention, and I look forward to your questions.